All right, lipids are going to be a little bit different than the carbohydrates you learned about the other day. And the reason is lipids don't really have an exact monomer and polymer, but they do still build these long chains of carbon, and so they are considered to be organic compounds. So just listen carefully when we talk about the monomers and the polymers, and you'll see why on your graphic organizer this one's handled a little bit differently. Lipids are fats. So fats, oils, waxes, any kind of fatty compound you can think of is a lipid. And they have the same exact elements in them as carbohydrates, just carbon, hydrogen, and oxygen. They're just in slightly different ratios. What we call the monomer in a lipid is the fatty acid, and it's not, again, technically a monomer. It's really just a building block that makes a larger lipid. There technically is no monomer for a lipid. The lipid polymer is this long, straight hydrocarbon chain with something called a carboxyl group added to the end. So, again, this is a little different than how you had the nice circular monomer of the carbohydrate. It just linked together. Lipids, like the ones in the pictures below, can be different shapes. Some of them have what we call tails. Some of them have 2 tails, like the ones right here. Some have 3. But basically what happens, and we'll go over this again, is that you have this glycerol, and then you have long hydrocarbon tails, 1, 2, 3 long hydrocarbon tails coming off of that backbone. So sometimes this is referred to as the monomer, okay? And sometimes these are referred to, when it's all put together, as the polymer. But technically, there is no monomer like there would be for the carbohydrate. The important characteristics of lipids that we want to know is they do not dissolve in water, which means they're non-polar. We talked about all the properties of water and how water is polar. Lipids are non-polar, so they don't mix with water. You know you can't get oil and water to mix. Lipids store energy very efficiently. Energy storage has to do with the number of oxygens bonded to the number of hydrogens bonded to the number of carbons. And lipids have a lot of hydrogens bonded to carbons, so they store energy very efficiently. They are more of a long-term energy storage, while carbohydrates are more of a short-term energy storage. Lipids prevent what's called desiccation. Desiccation, if you don't know what it means, let's write it down. It means drying out. They prevent things from drying out. In fact, most lotions that we use have a lipid in them. So if you turn over and you look at the label of a lotion, it probably has some kind of lipid listed, or maybe a wax. Think about things like shea butter. That's something that's very common in lotion. It's a lipid. Okay. Lipids make up the cell membranes of every single cell that exists. So we know that living things are all made of cells. Lipids make up those cell membranes. In fact, that's what this picture is right here. The cell membrane is a double layer of lipids. So each of these little units is a lipid. We'll talk more about why they're in this shape in a later chapter, but you should remember that lipids make up the cell membranes. Okay, so we have these characteristics. They don't dissolve in water, which means they're non-polar. We've written that fact down. We know that they store energy efficiently because they have lots of carbon-hydrogen bonds. They make up the cell membranes of your body and give you long-term energy storage, and they also help prevent your body from drying out. So there's basically 4 main types of lipid polymers. And again, remember, there's really not a great monomer example to write down here, so we're going to focus on the polymers. The first one's called a triglyceride, and it looks something like this picture that just popped up. Remember, sometimes people refer to this portion as the monomer and then these portions here as the polymers, but that's not really the technical definition. So we're just going to look at it as a large polymer molecule. I often refer to this as the backbone, and these as the tails that come off, and these tails can grow to any length. In the lab tomorrow, you'll build one of these triglycerides. It has 3 tails coming off of it, so we call it a triglyceride. It's a very large molecule, so we call it a macro, big molecule. It's 3 fatty acid tails, 1, 2, 3, all attached to a glycerol backbone. The glycerol is over here on the left. Examples of triglycerides include things that you're very familiar with. oils from plants, and fats from animals. So peanut oil, olive oil, corn oil, butter, lard. A second type of lipid polymer is a phospholipid, and a phospholipid could have been better named as a diglyceride because it looks exactly like this triglyceride, but it only has two tails on it rather than three. So this picture right here is a phospholipid. It has the glycerol backbone and has two tails coming off of it. Waxes are the third group of lipid polymers. Waxes are long fatty acid chains attached to an alcohol chain. So again, it's really that presence of that long fatty acid chain, just like we see up here, these long chains of hydrogens and carbons, that makes it a lipid. And the last major type of lipid we want to be familiar with are steroids. Steroids are lipids in your blood. This is when we talk about cholesterol. The steroids are a slightly different shape. They do have some rings in them, as you can see in this bottom picture, but they are still considered lipids because they do not dissolve in water. Now, the organic compound test for lipids is probably the simplest test of them all. It's the brown paper towel test. You've run a brown paper towel test in your everyday life if you've ever gone to a fast food restaurant and gotten a brown paper bag with French fries in it. And you know if the oil from the French fries gets into the bag gets a little bit translucent. To run the brown paper towel test, you simply put a few drops of your solution on a piece of brown paper towel. It is very important that it is brown paper towel. This will not work on some of the white paper towel we have in class. Then you simply wait for the solution to dry, and when it dries, if the bag or the paper towel is translucent, there is lipid in the test. The hardest part of running this test is remembering to run it at the beginning of the class period so that it has time to dry, so that you know you're really reading the correct results.